Welcome to computer lab number 3. So this worksheet has been posted. So in this there are around 6 questions which we are going to discuss. If you see this worksheet, so basically in this lab we are going to use all the tools which I just discussed with in the lecture. So the very first question is, use translate tool at XPESI and convert the following DNA sequence to the protein sequence. So basically we will be using XPESI, and how to use XPESI, so which is your SIB, which is Swiss Institute of Bioinformatics. So you can go on Google and type there XPESI. So it came XPESI. So this is XPESI is your SIB. This is your Swiss Institute of Bioinformatics, and you can click it. Okay. So now you are into the bioinformatics resource portal. Now you need to go and click here proteomics. Okay. So there are different kinds of tools on this, and we are going to use all the proteomics tools today. So what you're going to do, now we are into proteomics. The very first question in the worksheet is, you have been given the DNA sequence and you need to find out protein sequence. You have to translate it. So using the translate tool at XPESI, which means if you have DNA sequence, you need to make it protein. That's what translation is. So what you have to do, you have to copy this sequence. Okay, just copy this, and we now need to go to XPESI, which we already have opened. We need to go and look for translate. We need to look for the tool translate. So keep on going down, and you need to find out where the translate is. Okay, so here is translate. You will click it. So you will enter into translate. Now here is a window where you are supposed to put your sequence. So paste this DNA sequence here. Once you have pasted it, now click it, translate sequence. Okay, and when you do that, now you can see here you have got the result. Now in this you will see that you are getting so many different frames. So this is, as you know, that we provided the system, the tool, with the DNA sequence. The sequence of DNA, the DNA is double stranded. Okay, so this strand is going to, we have is converted into protein, and DNA is double stranded, which means both the strands of DNA can be read. So you can see here, this is 5' to 3', this is 5' to 3', this is 5' to 3', this is 3' to 5', this is 3' to 5', this is also 3' to 5'. So if you see these three, the DNA sequence is being read from one direction, and these three, they are being read from the other strand is being read. So as you know, there are two strands of DNA. These are one strand, and these another three are the another strand. But what is frame 1, what is frame 2, what is frame 3? Like if I go back and if you see that this is your DNA sequence. Now when the tool is going to read your DNA sequence, always when the DNA sequence is first of all converted into mRNA, and then we have already talked about these things, and then mRNA, the codons on the mRNA, they are going to be coded into amino acids. Now the information is basically coming from the DNA. Now if these are the, like, if this information DNA is converted into mRNA, if suppose I just for explaining it to you, if these are suppose this is your mRNA, then these first 3 nucleotides can be read as one codon. If these 3 are being read as first codon, then the another 3 will be read as second codon. Then the next 3 can be read as third codon. But so this makes one reading frame. Now these codons, they can be read like this also. Leaving C, it can be read as GGG. Okay, in the first frame, which I just explained, in the first frame CGG can be first codon, then GCC can be second codon, then AAG can be the second codon. The second frame, in the second frame, this G can be the first nucleotide, then the first codon is GGG. Okay, if this is the first codon, then the second codon is CCA. If CCA is the second codon, then AGA is the third codon. This is second frame. And also it is a possibility if first two nucleotides are not read and the first codon starts from the third nucleotide, then first codon is GGC. This is first codon. Then the second codon is CAA. Then third is GAA. This is third frame. So there can be basically 3 frames how mRNA can be read. Okay, so it can be, if this first nucleotide is being read, then this is first, this is second, and this is third. This makes one codon. Then fourth, fifth, and sixth will make the second codon. Seventh, eighth, and ninth will make the third codon. That is your frame number 1. But if first one is not read, then these 3 GGG is going to be read. So this will be your second frame. If first 2 are not read, then your codon is going to start from GGC. This is your third frame. And that is what this tool is telling you. It is reading both strands of DNA and it is giving you 3 frames for each strand. But what you need to do, you need to find out. So also in your mRNA, as you know that there is a start codon and then there is a stop codon. That is what the open reading frame is, which means this is the place from the start codons, which is AUG, which codes for methionine. These are all things we discussed in the first lecture. So AUG is the start codon, then UAG, UGA, and UA, these are the stop codons. So the protein starts from AUG, which means the first amino acid is methionine, and then it finishes. It can finish at any. So all these highlighted parts are showing you the open reading frames. They are basically showing you the peptides which are going to be formed. So what you can do, so in this protein, in this frame, it's showing you basically 3 peptides can be made. This is one peptide, this highlighted region. This is second peptide, this is third peptide. And in this one, this is your peptide, this is peptide. So what you need to do, all these highlighted parts are your peptides in different frames. You need to look for the longest peptide, and that is what the answer you're going to give. So you can see here in all of these, this peptide starts from here, goes up to here, it starts from here, goes up to here. I think the longest one is in 5' to 3' and frame 2. What you need to do, you need to copy from methionine, you will copy from here and you will go up to here. Okay, this is the protein which is going to be your answer. Okay, so you can just copy it from here and then you can go to your worksheet and here in the answer you can paste it. So this is what you are supposed to give in your worksheet. Okay, now let's move to next question. Using the reverse translate tool, let XPESI convert the following amino acid to DNA sequence. Now remember that I have talked to you about reverse transcription, all the microarray stuff when we did reverse transcription. Reverse transcription is when you can go back from mRNA to DNA. Now the question is reverse translate, which means you can also go back from protein to DNA, and that is what you need to do. So the DNA sequence has been the protein sequence has been given to you and you need to find the DNA. So you are going to copy this. Now you're going to copy this. So just copy this sequence, Control C, go back on XPESI. You can go back. Now you need to find the reverse translate. Okay, we now need to use a different tool. So you need to find here in the list reverse translate. It is here, reverse translate. Okay, so here is a window. You can just cut it. and here you're going to paste your protein sequence and then just say submit. Okay, and this is the answer given to you. And as you know that the amino acids, they are coded by, one amino acid can be coded by more than one codon. So here are some of the results. This is the reverse translation of most likely codons. This is reverse transcription base of consensus codons. You can take the first part. Okay, so on the basis of most likely codons, so this is your result. So you can just copy this result and paste in your worksheet. This is your result. So you can paste it here. Okay, this is what you need to tell me in question number 2. Question number 3. Do the homology modeling for the protein with this sequence. So a sequence has been given to you, and what you need to do, you need to make the protein model by homology modeling. So just copy this sequence, and now where you need to go back on XPSC. Okay, you need to go back on XPSC. So we can go back from here. Now what you need to find homology modeling tool. This is Swiss, we are going to use Swiss model. This is repository. This is Swiss model work space where you can make the models. You're going to click this. Okay, and you can say start modeling. Basically you have to click, you are not going to do anything. Okay, so you just need to know what to do. You need to paste your sequence here, and you have to see there are different things you can do. Search for the templates. If you just want to know the template, you can do that. You need to make a model. Okay, just click this. Now it is going to find the template for you, and on the basis of the template it's going to do the alignments. So first of all, on the basis of homology search using BLAST, it is going to find some proteins with a homologous to this, and on the basis of homology it is going to then align it, and then it's going to prepare the model for you. So see, it takes some time for it to make the model. We can go to our next question, and while we'll check if our model is made. So we can go to the next question. The next question is, find the important domains of the following protein using ProCite at XPSC. So this is the protein sequence. You need to copy this sequence, and you need to find out the domains in this protein. For this, what you need to do, you can so here our model is being made. So we can open another window here, XPSC, and you need to go on ProCite now, because you need to find the domains in the protein. So go on ProCite. So we will again go back here. We'll go into proteomics tools. Now this list should come up. Okay, click proteomics. Okay, I don't know why it's not working. Let's click one more time. We need proteomics tools. Okay, it is giving us. Now you need ProCite. So on the left side you can see the ProCite is here. Okay, and what you need to do here is the window. Quickly find matches of your protein to ProCite signature. So you can just okay, you can, the sequence which you have copied, just paste it here and then say scan. So it is going to tell you what domains are present in this protein. So let's go and see if our model is made. No, model is yet not made. So when you will be working for your own final project, you should remember that it takes for the homology modeling tool, the Swiss model, to make it takes some time. Okay, sometimes it takes a long time. Sometimes you have to start this modeling process at night, and the morning the model is made. Sometimes it takes a lot of hours. So you have to be a little patient with this. So we got the result for this. So this is the protein for which we want to find the different domains. So it is telling you these are the different domains. So you have to copy from here. See, this is telling you from here 12 amino acid 12 to 25. This is all information here, so you can copy from here and you can paste it in your result here. Okay, I'm just pasting one domain. There are a lot of other domains. You need to write down about all of them. Just copy paste there. So this is the first domain is called the crib domain. You need to have you have to copy from here all this thing. It is telling you the amino acids. It starts from 12 to 25 and all that. And then there is this protein kinase domain. It is telling you about that. And then basically you need to copy paste all this. Okay, so this is the third domain, which is protein kinase ATP domain, and it is making this nice picture for you here. So this is all information you have to copy from here. This is ProCite. And the next question is okay, using psort server, find the location of the protein with the following with the sequence given in the exercise number 4. So the same protein which we just had, let's copy that protein again. You need to find out for the same protein, you need to find out where it is localized in the cell. So for this you need to use psort. Okay, and in the worksheet I have given you psort prediction. So you need to actually go and basically I have given you here, if you come back here, psort2 prediction. So when you go on psort server, actually let's not go this server creates a little problem. Let's go to this server directly through Google, because it doesn't work through XPSE. So let's go on Google again, and in this you need to take psort2 prediction. This is psort2 prediction. Or we can maybe go directly to this. So this is this server sometimes takes some time to open. So basically what you need to do, you need to go on this website, and there you need to click psort2 prediction, and then there will be one window where you will be pasting your sequence of the protein, and then it is going to tell you in the end there will be information, a very concise information. It will be given to you that this protein is present 73% in nucleus, 8% in cytoplasm. I'm just saying all these values. I mean, I'm not saying this is the result for this question. Okay, this page is not opening. This page creates a little problem. So basically what you need to do, you need to go on psort server, okay, and from there you need to go on psort2 prediction, and a window will open. Just paste your sequence there, and in the end you get the answer there, and you need to just copy and paste that answer in the worksheet. So let's go to the last question, and if you have any problem with this, let me know. I mean, it should not be a problem. The only thing is sometimes this server doesn't open. Okay, the next question is to find, so this is the protein sequence given to you. We can copy this, and you need to find out okay, this is the protein sequence. You need to find out the transmembrane region in this protein, and for that you need to use TMPRAD. So for this, what we are going to do, okay, our model is mate. Okay, let's complete that, and then I can tell you what you need to take from here. Okay, for this you need to go to XPSE again to find the transmembrane region. We go to XPSE, Okay, go to proteomics, and now you need to go on the tool TMPRAD, which is to find the transmembrane regions. TMPRAD. So this is the one. Okay, TMPRAD. And what you have to do here, you can just put the name of your sequence, the sequence of the protein. And I mean, if you want to give some like query title and all that, these are all optional things. If you just put it here and say run TMPRAD, it has given you the answer. Okay, so it is on the basis of hydrophobicity plot, which amino acids in the protein are hydrophobic, because the internal core of the membrane is all made of non-polar tails of the phospholipids, and they're all hydrophobic in nature. It goes and looks for the hydrophobic amino acids, and on the basis of that it makes predictions that this part of the protein can be in the membrane. So here it is telling you, so here is your answer: possible transmembrane helices. So it is telling you, if you from inside to outside helices, this is the region which is found to be which can be predicted to be transmembrane, and this is outside to inside. This is the amino acids. So basically you can copy this result here, and you can paste it here in the worksheet, and this is your result. Okay, so the protein can be considered like, it can be, because if it's a transmembrane protein, if it's a membrane protein, as I told you, there can be some part of the protein which will be outside the cell, and some part will be inside the cell. So it is on the basis of how it is considering which part of the protein to be inside and which part to be the outside. On the basis of that, it is telling you these are the possibilities of the transmembrane regions, and that is what the answer is. So okay, for the model, let's go back and check our model. Second. So if we go here in this window, this is the model. Okay, so this is the model it has made for us. And the most important thing which you need to know, so this is the model. Okay, this is the model, and here this is also the model, and here are different choices. You can see this model in different ways. What you need to know, which is the most important thing for you, is the template. You need to know how the system, what kind of template it has used. So you need to provide this number. So you have to just copy it, and you are going to paste it here. Question, yeah, you are going to paste this here. This is the number, which basically means this is the template which it has used, and you can also show these results. How much is the you can actually copy everything from here, and also it is showing you, it is this template, this is the number given for the template, this is the description. This protein is outer membrane P4 protein. So you can actually provide this information, and you can actually copy paste this also and put it in the worksheet. So here you can copy paste everything. So that is the information you need to give in the worksheet, and you can also take the model. Okay, I hope you can copy that, but the most important thing is the template. So that's all your worksheet for the computer lab number 3 is. If you have any problem, you can always contact me, and I will be very happy to solve your problem. Thank you very much.